Thank you. Thank you for this opportunity, and welcome to my presentation. My name is Maciej Kandula. I'm from the Boku University in Vienna, and I'll be talking about a multi-layer network approach to data integration on an example application for patient stratification and survival analysis. So just a quick overview. I will just make a short introduction because we already heard a lot about this data set and about data integration in general. Then I will go into our method, into details. Then we will discuss some result comparisons to other method and to literature results. And then I will have some concluding remarks. Yeah, so a lot of hope is now being placed in the integrated analysis of measurements from different technologies. But there is a surprisingly little integrative research actually being done if you look at the statistics. So basically here you can see TCGA papers, papers that discuss, that analyze TCGA data for gene expression and copy number variation alone. And here you can see how many papers for integrative research on these two data sets are being done. This is basically from 2011 when the database was started until now. And this is not surprising because data integration is hard. And we don't really know how these different data sets interact and and it is hard to understand and predict such a complex system as a human being and how the disease affects us. And in meetings like CAMDA, one could think that actually everybody deals with data integration, right? Because it's interesting for us, but in the real world, as we could see in the previous slide, this is not really the case. And because of this, we need stronger methods like the ones presented by the previous speakers. And sorry. And this high dimensionality problem when we analyze genes alone is being reduced by the scientists where we analyze, for example, pathways. And there are different pathways and different databases, but basically this helps us abstract this the information from genes to actually some meaningful biological, say, pathways. And this, as I said, helps us to get rid of all these problems and helps to derive some higher level insights. And now I'm going to talk about my approach, and this is just to make you prepared for what you're going to hear. So we will talk about a higher level integrated information framework, network, that will comprise of patient networks, pathway networks, and following, as we know, pathways are actually gene networks, so this is another layer of pathways. And here the layers are derived from our domain knowledge. Yes, so this is what we are aiming for. So each of these networks are patient networks. They are derived for each different data type. Not necessarily only these two, but this can be any data type. We produce a network of patients. And in the end, we want to get an integrated network of patients. But in order to get it, we first need to have these for the specific data types. So let's now only focus on one of these networks to just like spare the space on the slides and make it better understandable. So here the nodes, so the black dots basically are the patients, and we have a weighted similarity score between the patients, and I'm going to discuss what they exactly are in the next slides. So let's focus only on 3 patients, because again, this is just going to be easier for showing it in the slides. So we have our patient network, let's say for gene expression and for these 3 patients. Oh, sorry, it went too quick. And now each of these patients is actually a network on their own. And what this network subsequently is a network of pathways that are basically derived for each patient and each data type. So here, again, the connections between the pathways, between the nodes in our network, are again weighted similarity scores. Sorry. And how these are derived, they are based on this kind of framework. So basically, we supply our network with differential effect analysis per patient. This is per patient, and with we integrate the information from our differential effect analysis with CAC database information and with Go terms information, and we basically link the pathways, so the blue dots, with each other through CAG pathways, which hold the same genes or some of the same genes in common with our pathways. So in the end, what we get is a network of pathways weighted by the gene expression of the genes similar to these pathways. Yes. And then the next step here is actually fitting a stochastic block model into our network here and looking for a model, so the stochastic block model, that best explains the network's internal structure. So basically, we group the nodes of pathways. So what we next end up with are these separate networks per patient and per specific data type. So what we now do, sorry. So what we now want to do is actually link the patients through a similarity score that I said we are gonna learn what it is. Yes, so what we are doing here is we get a specific clustering for patient 1 and specific clustering for patient 2. And now using the variation of information metric, we compare clustering of the first patient with the clustering of the second patient. So the variation of information is looking at the entropies between the clusterings and the mutual information between the clusterings. And we are getting basically a distance measure, which is a metric. And this is an important point. And now we are getting at our larger network back here. So this is for gene expression now. And now we do the same for copy number variation. Again, we have this different networks for each patient. Now we look for the distances between them, and we derive the next network for copy number data now, or methylation data, or what you have. And here we have our two networks, where each network is built for a specific data type. And now we can either perform further calculations in this patient-patient network per each data type separately, or as this is basically now a patient-patient network with the edges as a vector of multiple VI values, right, for each data type, we could do some kind of integration. So as VI is a metric, we can perform any calculations directly on the values that we got without any special normalization or anything like this. So what we are doing here is integrating the VI values of all the data types per patient by averaging the VI, the variation of information. And so now what we get is this integrative network that I've also shown in the beginning. And naturally, we also, as I've shown, obtain such a network per each data type. And for survival analysis in particular, we can now try to, again, group the patients basically here, the nodes. So here, I chose the gene expression network here, so the black network, just because it's easier to show with the different colors. But basically what we are doing here is fitting a stochastic block model to again explain the underlying structure in the network. and we are getting this different groups of patients. And this is what we are going to learn later, but this could be, for example, a group that has a high risk of dying, or low risk, or something like a moderate risk. This is all that we can also learn from then later looking at the clinical information, which of course we are not yet using in the network. This is something external that we can later compare our results to. So with such a result, we can now perform survival analysis. And first, what we do is we show that the found groups of patients are stable by removing a patient from the network. So now we remove a patient from the network, and we see that our approach will classify the remaining patients in the same groups as it did before we removed the patient. And we can see that it's highly accurate, almost 100%, with the mutual information between the groupings. This is average again, almost at 1, and 1 would be a perfect similarity, and very high correlation as well. So next we compare our new approach with the best results found in neuroblastoma literature. And so the results for literature are in the last column here, and we are showing here log to HR because they are just easier to compare between different approaches, as the HR of 1 would mean that there is no effect. So it's harder to compare values below 1 and with the values above 1. So we just do here a log 2 of the HR value. So basically these are the best results for literature, for ACGH, and for microarray, and we can see that we outperform with at least with log 2 HR. So with hazard ratios, we outperform the best results from the literature. And with our integrative approach, our grouping actually highly outperforms the literature, the results found in the literature. And let me just mention that what we are focusing here on are the 145 patients that have common data for microarray, RNA-seq, and ACGH data. These are 145 patients that have data in all these 3 data types. Yes. So next, we compare our method with the already mentioned SNF method. And here we compare the best results obtained by SNF and best results obtained by our method in an approach where we test different numbers of clusters, because by clusters we mean in how many groups should the data be split. And here we show only results where, of course, where all the found factors are significant, because if the found factors, so one of the groups is insignificant, then the result is basically not really clinically actionable. And here also worth mentioning is that the Cox regression is done with a 5th correction, which is important for this kind of data where we have basically, I think, 80% of the data is right censored, so we don't know if what happened to the patient after the examination, basically. And the big score is the Bayesian information criterion, and basically the lower it gets, the better the performance of the model. So we can see that our integrative approach outperforms SNF, both on the 2 clusters as well as when we have more clusters, and this is both on the big score and also on the log to HR scores, or HR basically. So here, this is just to have a look at what our groups look like, if they make sense or not, and we can actually see that yes, like this is for 2 clusters and for 3 clusters. So in the 3 clusters, group number 3 comprises solely of patients whose survival time is censored, and this is reasonable and not unexpected, but it would be a huge problem in Cox regression without using the 5th correction. The results would not be reliable, basically. And this has been proven in literature. And now just to have a look how our results how our groups also compare to the clusters by clinicians, the high risk. So basically, we can see that in the 3 clusters, the first group is basically of a very high risk. Second would be, for example, moderate, we could call it. And the third one is basically low risk. And here, we also next now combine the clinical labels with our integrative approach, and we can see that the model gets actually even much, much better here. So just to sum up, we developed a method that performs on different layers of networks, and the best models are obtained actually when we combine the molecular data with clinical information. And thank you. I would like to thank my supervisor, my PhD supervisor, David, our collaborator from States, Eric, and especially our smart and very highly motivated student from India that's helping me pre-process the data and work on this project. Thank you very much.
Great. Can you give some intuitive explanation of why this method of working first with the patient networks and then going up works? What kind of noise does it cleanse, or what's the mechanism for the analysis?
Yes, this basically helps us abstract the high dimensionality of, for example, looking at only genes into higher biological information, so to say. So I would say this works a bit like looking at the pathways instead of looking at genes alone. Not sure if I answered your question. So do I need to weight the differently with
you use the average?
No, I use the average only to integrate information from different data types. You could test something else because, as I said, the VI is basically a matrix, so you can perform any calculations on it that you wish to. But we have shown that this simple approach actually works pretty well, or at least better than single track data.
Okay, thank you.
Welcome.
Okay. I have 2 technical questions. So definitely your method is very interesting. Yes, it's passed through several, different methodologies, data structures, cluster analysis, classification, statistics. So I have 2 questions. So the data structures, would you explain what sort of data structures you're using in your model? So let me say, because before starting to integrate data, you should define structures. So the second question is, you're using only Bayesian information criteria for statistical significance. So do you try, Bayesian information is a little bit more weak than, let me say, maximum likelihood. Do you have you tried to use some another statistics? This will be used like a cross-validation for that.
So I only tried AIC as well, but the results looked almost the same. They just, I mean, the numbers differed slightly, but the ranking was the same, and also the differences between different methods and different approaches were also relatively the same. And the first question,
data structures, yes, because we are going to integration, towards integration of the data. So what sort of data structures you're using? Technically, how do you prepare your data?
How do I prepare my data? Yes. So we do standard pre-processing so we need at first to perform differential effect analysis. So this will be dependent on the data type that we are using. So for RNA-seq, we were using VUM-TMM and Lima. For microarray normalization, I think quantal normalization with and then Lima. And actually, ACGH data we analyze as well like this. So using Lima to find the differential effect. So we treated the patient of interest as the cancer samples, let's say, and then all the other patients as the normal samples. And then we looked for the differential effect between the patient of interest and all the others.
Okay, thank you.
You're welcome.
Any more questions?
Could you potentially translate this data to a new cohort or incorporate extra patients if you got another set from another center?
Yes, I mean, I tested this also on BRCA data initially, and it worked quite well as well. But this is something that we will be doing next. And regarding adding additional patients here, it seems that clustering, I mean, reclustering of the patients is very simple. So we can add a patient and then get, so to say, new groups. And as we've tested, if we remove a patient, then the groups of the others actually stay the same if we redo the clustering. So basically, adding a new patient should not theoretically negatively influence, let's say, grouping of the others. So it should work pretty neatly, actually. And also from the models that we get, from the SBM models that we get from clustering the patients, we can actually derive a grouping of a new patient directly by doing a regression on the model that we got, actually.
Yeah, sorry, you may have explained this, but I joined the talk late,
Sure.
so I apologize if you already explained. So I noticed that you had some results for 2 clusters and 3
Yes.
clusters. Do you find the optimal number of clusters, or you try
Yes.
several?
So we tried clusters between 2 and 9, and we basically saw that at some higher numbers of clusters, that even though the BIC might get a bit lower, the hazard ratio also gets lower, but you are starting to get lots of factors, so lots of these groups in your clusters that are insignificant.
If I want to apply the method to a different data sets, it will say the best number of clusters for this new data set is X.
Yes.
Thank you.
You're welcome.
Great. Yeah, good discussion. Our last speaker for the session is
Thank you very much.
Danke.